We'll start the recording. So, happy Wednesday.
Hi.
Hi. So what we're going to do is today we're going to continue our conversation about microbiology and our microbial world. We're also, I know some of you had emailed me and asked about a test today because we're trying to do for a multiple choice test on lecture notes. But I didn't want to do it today because I was going to cover stuff, finish some stuff up before we did it. So let's just don't worry about that today, and maybe have from today a lecture quiz on Monday, and on Monday we'll discuss when the next test date would be. Y'all are working on a take-home test for Chapter 10, and if anybody started that, maybe you can verify with your friends that everything is straight out of Chapter 10.
Yeah.
So good, and take your time with that. You know, there's no real hard due date on that, but just so you don't get too far behind, I would kind of suggest that you get them into me probably by the end of the month or the very latest, maybe that first week in April. Okay? Yes.
I just have one question. You told us to paraphrase it, but like some of the answers on that are like very like head on.
So they're very simplistic.
Yeah.
That's
So, like,
okay. I get that. You know, guys, y'all think I'm so mean. You know, I really, when I say, don't give me a textbook definition, and y'all are like, oh my God, there's no other way to say it. So, look, that's fine. That's fine. If I can just know that from your simple statement that she would have absolutely, Brandy would have absolutely understood what this meant. You know, that's what I'm trying to see.
I just like, for some of them, I was just like, if I change the word, it's not going to mean.
Oh, yeah, no, I get that. Please don't think I'm that crazy. So we know we've got that going. So today we are going to continue on those. And in lab today, it's just a very brief thing. And I want all my people going because it's only going to take a few minutes. But you're going to, today, we'll hook up the back to incinerators real quickly to get those hot. And today you're going to have a couple of sheet blood agar plates, and I'm going to give you some sterile swabs. And using sterile technique, so you don't contaminate your swab with something on your fingers or on the desk or whatever, you're going to go to any body part you want, and I want you to do 2 body parts. So you're going to be inoculating the plate. So today you're going to be and you don't have to share with anybody what your body part was that you did. So no disrobing in the lab. So that just will not be. But you can go to the bathroom if you want to. If you don't want to see somebody see you sticking it up your nose or whatever, go to the bathroom, get your specimen, carefully open the thing, and you can put it back in there, right, and bring it in and then do your plate. So anyway, does everybody remember, just real quickly, since I have you all here sort of listening, do you remember that you inoculate close to the edge, about a nickel size, dime to a nickel size, rolling the swab easily? Then you can throw that away, the swab. But you're going to sterilize your loop and streak it really, really well in that first third of the plate. You remember that? Then what do? You sterilize your loop again. Make sure it's not scalding hot. Let it cool. Let it cool. Go into that little area and go into it at least 3 or 4 times. And then you've got your second quadrant. Sterilize your loop again. Let it cool completely, and do that until you've got this nice. Now, some of you all saw that you didn't maybe do that sterilization thing too well because you didn't have any real isolated colonies, did you? And I'm going to tell you that you guys, where you are going to be getting cultures from, most of the places you're going to get them from, there's going to be a lot of different types of organisms that are there that are going to grow. So you really want to do that technique. You want to use that technique so that you can see isolated different colonies. Fair? So all of that being said, it usually only takes a few minutes for you all to do this. Now on Monday in lab, you're going to be looking at those plates, and I'm probably going to see how they look when I come in tomorrow. I'm going to pull them out, put them in the refrigerator, and get them to sort of stop, or at least put them at room temperature. But tomorrow, when I'm looking at them, depending on kind of how I see, I think I'm going to probably get you all to try to isolate colonies to new plates, but you're going to be doing some quick identification, but you're also going to be doing susceptibility tests. So any of you that have looked at Chapter 10, you know that the Kirby-Bauer method of doing susceptibility test is that you take pure isolate, a pure organism, and confluently put it onto a plated media, and then drop your antibiotic disc on the thing. But we're going to be doing that next week, so we'll talk about that then, okay? Use good technique today so that you'll have an interesting time next week. Any questions for me about that? Okay. All right, so how, everybody's good, right? Did anybody bring anything in going on in the news that they wanted to talk about before we get started with some of the notes? No?
So, this doesn't really have to do with microbes, but I saw that they're gonna, I know you have to have like a brain biopsy to tell if you have Alzheimer's, but they're gonna start looking at people's eyes and the vessels. So you can see exactly.
That's, thank you for bringing that up. I saw that too. So could y'all hear, Brittany? So a confirmatory test for Alzheimer's, unfortunately, has been brain biopsy. So obviously, most people who are alive don't let you do that while they're alive. So that's done after death. With the presumptive diagnosis of Alzheimer's for those years that the person's been treated. But this is exciting that there might be. Can you tell me what they're looking for? Because I didn't see what they were looking for. Looking in that. So it was just the blood vessels, so that they're looking at that anterior surface of the retina where we can directly see blood vessels. And did they say what they were looking for? Because I did see that they wanted to look in the eyes, but I didn't hear what they were looking for.
I saw like the flashlight, like I see it's just a quick examination. I'm assuming it's like the lack of vessels.
Yeah, it's going to be interesting though, and I wonder how. I wonder like if the false positives or false negatives, you know, like what those numbers are, but that's going to be nice. That's going to be nice. Thank you for bringing that up. And I do want to hear more about it. So if you hear about it, y'all bring that up and bring that in. I think it was Sarah, the zombie disease. So that was in this health and science thing too, talking about this chronic wasting disease. So it's funny how you see things right after somebody brings them up, right? So they are really talking about this, and I don't know about y'all, but I'm not eating wild deer or elk or whatever. But honestly, just I think we had already said, but it looks like Virginia there is very rare, but in the Montana, Wyoming, it's a lot.
It's in like 24, 25 states, and then
25 states. Yeah. And it's prion, which you all know is really difficult. And the thing is, it's so slow in affecting, the animals that it's just a slow progression. But I think unless you know how meat is being handled this day and age, there's no real reason to risk it, and whether meat has been contaminated with cerebrospinal fluid or any nervous tissue. My other son, we were talking about it, and he was saying how, when hunters kill these deer and stuff, a lot of times by how they're shooting and whether it's bow and arrow, what it might be, they can nick that tissue. And then it would have, even if they're handling it with what they think is most pristine types of techniques, it could be contaminated with this. So, I don't know, just something to think about. Yeah.
Somebody I knew got sick, and I think, I don't know what they got, it messed with their vocal cords, and, it tore one of his vocal cords, and
I didn't hear the first part of what you were saying.
He got sick, and I don't know if it was a cold or something, but it tore one of his vocal cords. So he can I don't know, heal or will have a voice.
I didn't hear the first part.
Sick. He was sick.
Yeah. Who?
Somebody
she knew.
Somebody you okay? So you're saying I was like I'm missing something. Lord have mercy. Okay, so are we good? Can we start again? So Molly, you're saying that there's somebody who has had a sore throat? Is that
he just got sick. I don't know what it was, but he got sick and one of his vocal cords tore, and now his voice is all messed up really bad. And he's been to 3 doctors, but they said they don't know if his voice will come back or if his vocal cords will heal enough.
wow, that's horrible. Did they think there was an infectious process that happened?
Yes.
So they think this was in consequence of an infection that had somehow damaged those cords. Yeah, that's horrible. I've seen people who have had some permanent damage to vocal cords that really, it ended up not being an infectious process. It had to do with reflux and the constant irritation with acid that can happen from the stomach that's coming up the esophagus and leaking over into the trachea that can actually end up damaging vocal cords and causing people's voice to be really, really, whatever. Well,
And like he was sick for like 2 weeks, and they just thought it was like the cold, and he got treated for it.
so he really did have some sort of infectious process that did it, not chronic pain. Well, I hope for the best for that person. Look, guys, and so this anti-vaxxers impact, we just continue to hear about this, don't we? And on social media, so much stuff. I know y'all are hearing both sides. Y'all are seeing both sides, aren't you, on social media? Do you ever weigh in? We all have restraint, I guess. No, you don't weigh in on the social media sites when you see pro or con for
I can't
vaccine.
stand to read it.
Oh, so you just skip it? Okay, out of sight, out of mind.
Well, no,
All
I
right,
read
then.
it to a certain extent. Like, I get to the point where I get really mad, and then I just say, you know what, it ain't worth my time.
I saw
Okay.
where a little
Okay, Dakota. What'd you say?
I saw where a little boy got tetanus out west and he died and he had like over $1 million. Like he wasn't vaccinated, over $1 million with medical bills, and if he would have gotten a shot.
Yeah. So,
Yeah, it's like the first child case that, and he died. That they've had in, I think they said 30 years.
well, tetanus. There are a few cases each year, but typically you can treat it
Well,
because
in that
early
case,
enough.
yeah.
Oh, in that particular case. And it's rare in this country because we do have immunizations. In other countries, it's still a significant concern because they don't. But we are we take a lot for granted in this country, don't we? Until we've lost it, and like lost protection from herd immunity when enough people in an area are not getting immunized, or when whatever. But science we talk about as being this idea about being able to test things in the natural world, and it's so limited to that. It's limited to that. You can't really, don't even pretend to test things or know anything about metaphysical or other types of things. So it's limited to the natural world. But in science, we've made incredible strides with enough testing, enough data that can give us confidence in certain things. So there's not, it becomes, okay, this is going to sound, it becomes not a debatable issue because the data is there to support the millions of lives saved from immunizations. And so, so when you do see misinformation being spread, maybe it's a responsibility to try to help alleviate concerns. Nobody intentionally wants to not get their children immunized because they're just trying to stand on one side of an issue. We really have fear. There's really fear. It's true. It's fear. It's real for them. It's so real. And so it's part of people in medicine. It's part of your responsibility, really, to try to help, to support, and to show the data that supports this idea that we have come so far from my big mama, but so far from her, who only expected half of her children to make it to 5. She only expected that because that was the reality. But we have forgotten that. And the reason that was the reality is because they didn't have immunizations or antibiotics. So we have really forgotten that reality. But that could become a reality again with fear-mongering and lack of support, because people are truly afraid. People are motivated by what they do for those reasons. They're not doing it just to be obstinate. Yes.
I'm doing a project on like vaccinations and immunizations like worldwide in like different countries, and I read so many articles about how like different places like Costa Rica and just other places other than the United States like spend millions of dollars trying to get everybody vaccinated. And, like there's so many people that want to get vaccinated. And there was one article I read, and it was like the United States, like people who don't get vaccinated, they take it for granted because like other countries like struggle with like vaccinations.
Yeah. So yeah, and that makes you sad too. People who would want their children protected because they've seen their older children die, and they're convinced now. They're convinced, and they hear that there could have been a chance to prevent it or whatever. So they're convinced and they want it, but can't get to it. It's not available. Yeah. So we're better. We've made such great strides, and we just need to continue to be better. And not like out of sight, out of mind. Okay. I'll read the code. All right, so look, and this other thing, and I'll leave it here so people can look at this if you want to. We talked about our microbes that live in and on us, and how those microbes actually kind of train our immune system. We know that they do. And I'm learning more about it all the time. This particular article is really fascinating. Again, it's data driven, because enough data can really speak to you. But how childhood infections and antibiotics may increase the risk of mental illness in children and people as they age, because the balance of normal flora in the gut, they call the what do they call the gut flora again? Are you talking to me? What? That's the Robert. The gut flora, the gut microbes. Do they call that like the second brain? I think you told me that.
Yeah, the second brain.
The second brain. And that balance and keeping that balance. Certainly when we take antibiotics, we can't talk to them and say, okay, only go toward my infection and leave all my normal flora alone, because they don't listen to you. So getting treated for antibiotics and save millions and millions of lives. But it's something to consider, to use them wisely, use them appropriately as far as like the right ones for the right illnesses. And this is where the microbiology lab and diagnostics come in. And great strides are being made to get to those IDs quicker than we ever did before. So, we just hope that works that way. Any questions about this? So if y'all want to look at this and see how that could actually affect your gut flora, affecting your other brain, then you can take a look at this and some of the things that they're noticing with very specific types of enzymes and proteins that are affected, and how they think that some of these enzymes and neurotransmitters being affected by that are contributing to some of the psychosis. All right, so that's all I had. And thank y'all for bringing up the things that y'all did. I am going to put a date thing today for y'all to do a study guide for today's lecture. And the reason being, and I'm going to fully admit this, is because I'm going to use them for your next test. Not only your lecture quiz for Monday, but I'm going to use them. I need them for your next test, just to keep myself honest and which questions I'm writing for the next test set of information. Good? So
will this chapter 11 be on the next?
Yeah, so we're going to do this chapter 11, and then we'll stop with the information, and that will be on the next test.
So 5, 6, 7, 8,
And 11. That sounds like a whole lot, but it's just what we talked about, right? That sounded like a lot of chapters. Really? What chapters are they?
5, 6, 7, 8, 9.
Yeah. Is that okay?
5, 6, 7, 8, 9, 11.
Jesus Christ.
Okay, so all right. So a lot of this, let me say this, are y'all sensing already, and we've only been together for like however many weeks now, but are y'all sensing that a lot of topics reoccur and a lot of the language is just reoccurring? So I hope you don't feel like it's all just brand new to you. Still, you might, but I hope you don't. Anyway, we understand that we live and we coexist with these microbes, and we depend on them. So we're actually depending on them. But sometimes we do know that certain So certain microorganisms can take advantage and become opportunistic pathogens. And that's in here too, but that's something you already know about. The Human Microbiome Project, haven't we already talked about that? So we've already talked about that. Just make sure that you do understand that we've been studying this now for more than a decade, and so much new information is coming out all the time. It's really hard for us to keep up with it in the field. But anyways, but this is information we want to be paying attention to. So, just the whole idea that as far as our human cells go, we said really the traits and genes, our human cells, we have 25,000+ whatever genes with this many, and I don't ask you those kind of specific numbers, but way more of our microbes, way more encoding genes, right? So how that balance that we have, this makeup of this balance, influences our overall health. And I really do think that we're going to see so many other areas that it affects, not just our immune system response and how that imbalance might be leading to more autoimmune diseases and leading to maybe even mental health disorders. Our human condition has greatly improved. Our lifespans have almost doubled in a very short period of time because of modern medicine. But at the same time, there's some things we're doing that we know that are probably, how long do we want to extend our lives, right? We might want to ask that question. But anyway, there's some things we're doing that we know are just so counterintuitive to us as animals. All the preservatives that we have in foods we know affect our normal flora, right? And so that's why they say that, you really want to limit those in your diet. We know about epigenetics now and how our environment is so influenced about, it is so influencing our own DNA makeup. So there are a lot of things that are a little bit unnatural, like the fact that we're not outside as much. And y'all are hearing all over the place about how kids that live in a greener environment, that are exposed outside a lot, grow up to have less things like depression and all kinds of other illnesses. So, but we have so many people now, and society's changing in such a way that really a lot of times we're in rooms like this, aren't we? No outside light, no whatever. We're staying up all night, we're not sleeping. So all those things. So we talked about all this before. We know that most areas in our body are colonized. And when I say colonized, what does that mean? That just means there's microbes living there on that. And so we know most of them really are. There are some sites that we think of that really there's nothing. So they're considered sterile sites. So certainly like the central nervous system and really the bloodstream. Yeah, we can say the bloodstream, and we can even say urine is a sterile body fluid. But is there a portal of entry for microbes to come in? Yeah, we do know there's portals of entry. So what we think about is even sometimes these areas that we know are sterile end up having what's called transient microbes in them. Usually it's bacteria, but they're transient, meaning that they're only there for a short period of time. They're not actively growing because your immune system gets rid of them, but they could be detected for any short period of time. But there are some places that we think of still as being sterile. So certainly those sites you would want to keep sterile. Okay, so some things that weaken host defenses. Does this all make sense to you? Do you think a patient's age matters when you're considering whether they would have an infection or not? Absolutely. So we tend to think the very young and the very old, don't we? Mm-hmm. And we said there's some things, though, that we know that actually target that young, healthy group. And what are some things that we say can target a pretty serious infection that would target a young, healthy adult? Yes, that Neisseria meningitidis one. So there are a few things we know that can wipe out the prime things, but most of the time we're thinking about the very young, the very old, and that's what we know to be at risk. Surgery, anything that's insulted somebody, trauma, surgery, transplant, certainly any of those things. Cancer is it's considered a wasting disease. And so because of that, because the cancer cells are usually pulling things from the normal cells, then we know that this is going to be a problem too. Any chemotherapeutic agent even though it has a therapeutic use for whatever you might be treating, is also at the same time typically going to cause immunosuppression. So again, we talked about all of these, and I think they're intuitive, don't you guys think? Right? Now, with pregnancy, what we really worry about with pregnancy, is it always just the mother that you're worried about, or can some things cross the placenta? So some things can cross the placenta and become teratogenic. You have that word. What does teratogenic mean? What does it mean? Do you have that word? If you don't know that word, let's write it down. Teratogenic. T-E-R-A-T-O-G-E-N. Teratogenic. T-E-R-A-T-O-G-E-N. I-C. Teratogenic. A teratogen is anything that can cause harm to a fetus. So something that is teratogenic is causing harm to a fetus. You see what I'm saying? So anyway, that's what that word means. And we know that there are a lot of infections that can cross the placenta, name some, and harm the fetus. HIV. Now, HIV is the virus that crosses the placenta. So HIV could, it doesn't always, it isn't a given that it's going to, but it can. So what can reduce the risk of somebody who's HIV positive sending that to their fetus? If they're being treated, if that person is getting treatment. This is why it would be really important that somebody knows their HIV status before they're deciding to get pregnant, or before getting pregnant, or at least while they get pregnant, and then they can go on treatment, because it reduces the number greatly of how it can be. It can still infect the fetus, but it reduces the risk of it tremendously. Worth doing. Yes.
I seen on the news where it was the second person with HIV.
Yeah, that was on the news. That's huge too, isn't it? That's really huge. So did you all see that? Where thank you, Molly. That's huge out there right now, that the second person has what they think is a confirmed clearing of HIV in their system. Now, did you read any additional about how it happened?
Something like the bone marrow and everything. Like they got a transplant because they had like cancer or something.
Exactly. Both of the people, and they're the only people we know that have gotten cleared. Both of the people that it happened to had cancer that was a serious enough cancer that they needed bone marrow transplants. So does everybody understand that we can't use that as a treatment for HIV, bone marrow transplants? Because that will kill you. So these people were getting bone marrow transplants because their cancer was going to kill them. So they got the bone marrow transplants because of their cancers. They survived the bone marrow transplants, and then no longer was HIV detectable. So in those two patients, it really does seem that way. What we know, it's called viral load, is that right now it looks like they're cleared. But it could be that it's so low that it's just not detectable at this time. But it's looking like they're cleared. This is the beauty of this, though, is that it's given insight into maybe what could be done that wouldn't require a complete bone marrow transplant. It would require just changing the receptors on the person's white blood cells, because these people who got the bone marrow donor transplants, the donors had a different receptor on their white cells that wouldn't allow the virus to enter. So instead of having to give them an entire bone marrow transplant, now research is going to go to maybe changing a person's own receptors. And that could maybe be done with one of the ways they're looking at is recombinant DNA technology. Do you all know what that means? Yeah, you do. By inserting foreign DNA recipes to change the host recipes for that trait. So if we could change those receptors on a person who's HIV positive, change the receptors that are allowing the HIV virus to invade those white cells, then that would be a way to potentially treat. That's exciting, isn't it? So that is exciting. It's something for us to be looking at. So, right. So what we have always thought in the past, and again, new information all the time now, is that a fetus was really essentially, except for some things that can cross the placenta, like measles, mumps, another reason not to have outbreaks of measles and mumps, because the unborns can be at risk if their mothers become infected. Measles, mumps, rubella, CMV, cytomegalovirus, HIV. There's some things we know that can cross, that there's a risk of causing teratogenic damage to the fetus. So there are a few of those things. But we've tended to think about the fetuses as being sterile because they essentially are isolated. They're sterile in this amniotic sac that is got amniotic fluid. They're like these little water creatures, you know. So they're in this, that's considered to be really sterile. So unless something breaches it, or it's one of those rare kind of pathogens they can cross, then we tend to think about a fetus as being in a sterile environment until the amniotic sac breaks. This is why when somebody's water breaks, y'all have heard of the water breaking, it's the amniotic sac breaks that's filled with water. And so when that water breaks, a fetus will have to be delivered within 24 hours, because there's now a open system from the vaginal, the birth canal, into the amniotic sac, and that's just going to be like an open culture plate for microbes. That's not that wouldn't be a normal environment. That fetus is going to have to be delivered, right? And that's why they're also, I don't know if y'all have ever, I know some of your moms, so you know this, they're really careful about how many times they check your cervix for dilation, because every time you got somebody, even I don't care what kind of gloves they've got on and how much iodine they put, when they're checking your cervix to see how much you've dilated, they're going in and checking that. They know, they limit how many times they do that, because they know every time they do that, they could be introducing something to the fetus. You see what I'm saying? So okay. But we know that this vaginal flora, we've already talked about the idea too, that this, especially Lactobacillus acidophilus, this is a predominant flora that we know can help the babies to digest milk. So we know that this is all normal, isn't it? So much so that babies that are being born now in clinical facilities through C-sections, they are trying to swab the mother's vaginal canal and creating a, well, I don't know what you want to call it, not a solution, if you will, and actually rubbing the baby, the C-section baby, with it, because they're just hoping to somehow, you know, get that same sort of experience. But of course, do we know what we're doing really when we're doing that? Not really, but anyway, I don't think it's a bad idea. So you're seeing some of this, and we know that this is all important because it's one of the first steps of the baby's immune systems being exposed to normal flora. All right, I've already talked about that, Hannah. All right, so we have a definition. See what I mean about this chapter, having stuff you already know. Don't look at the numbers of chapters. It's just, okay. So a pathogen we had defined already. I had to do that long ago. But this microbe that is going to actually potentially cause infection and disease. Disease, on the other hand, if you never really thought about what that is defined as, not all diseases are infectious diseases, but we're concentrating on the ones that are infectious in this class, right? And when we talk about infectious, it just really does mean that they're caused by microbes and their products, and truly they could be communicable. And when I say communicable or contagious, what does that mean? They can't. Some of them are very communicable, contagious, and some aren't. Some are not as easily spread. Is HIV easily spread? No, really. It requires a direct fluid contact, and it requires direct certain fluid contacts, doesn't it? So, you know, we know it doesn't live on inanimate surfaces very long. It doesn't. There's not even enough of it in saliva to be infectious. So we know that casual contact and whatever, this is safe, you know, with HIV. It's not with all things, but it is with that. So for each of these diseases, we kind of know. And I've talked to you about the virulence factors or pathogenicity, as far as understanding that some things will actually be more harmful than others. I've given you a definitions for true pathogens, haven't I? So see what I mean? So this is just a review, and I hope you all are feeling like it's very comfortable information. So a true pathogen is going to cause disease, and it doesn't matter. They should never be present, right? So they should never really be present. And then we talked about opportunistic pathogens. And when we think about those, obviously these are the ones because the patient is immunocompromised, and it might just be because they are their age, right? Or it could they very young, very old, whatever reason they're immunocompromised. Then you've got this organism that's going to take advantage of that and cause disease. So y'all have that in your notes. You have virulence. You know what are some virulence factors. When we talk about portals of entry, certain microbes can only enter in certain portals. Some, require other portals. So I think I had also talked to you about that. I said if you had cold viruses, rhinoviruses, adenoviruses, do you remember me talking to you about those groups? They're on your list. Rhinoviruses and adenoviruses cause the common cold. You can swallow thousands and not get the cold. But one on your eyelid or in your nasal mucosa, and what? You're going to get sick. If you haven't been exposed to it before, you're going to get sick. Exogenous means that these microbes are coming from an outside source in the environment. And endogenous is going to be meaning that they came from they had been silent, part of normal flora, and then they're going to come from within. Those are just basic definitions there. What else do I want to tell you? Let's see. I think I had mentioned infectious dose, but if I had not, make sure you write this down. It's called the ID number, the infectious dose number. For each microorganism that y'all have on your colored sheets, there is a known infectious dose number that has to be present. So look at how different it can be. Rickettsia. Does anybody know what rickettsia causes? What'd you say?
I don't know if it's pronounced rickets.
No, but that would have been a good guess. Rickettsia is a spirochete. It's a bacterium that causes Rocky Mountain spotted fever. That's on your chart, so now y'all are going to have it in your notes. So you're going to make sure you highlight it. Brandy's getting her chart out and highlight it. Okay, so rickettsia causes Rocky Mountain spotted fever. And how does it take much to make you sick? No. And by the way, have y'all heard of this disease? So some of you've never heard of Rocky Mountain spotted fever, really? Yes, it's a tick-borne illness that is in this area. You can look at the CDC site where it shows you where most of the cases occur. It's been in this area, and it has a significant mortality rate if not caught early. Rickettsia. Y'all see it? It's a bacterium. So it's a bacterium. Rickettsia. All right, so y'all might want to just do these in your notes and then later go through the page. But anyway, y'all already have tuberculosis, don't you? What causes tuberculosis? Mycobacterium tuberculosis. And what is it? What group is it in? Bacteria, right? So the good news about these, even though it takes very little cells to make you sick, and they have high virulence factors, the good news is that they're bacterial. So what does that mean? Why is that good news? We can treat them with antibiotics, and we need the antibiotics, saving lives, right? Saving lives. So this is actually a really good thing. Giardiasis, Giardia lamblia, it's a protozoa. Do you have that in your notes? You have it. Have you already heard this one before? It's a protozoa. So it's a protozoa, Giardia lamblia. These colored sheets might come back and haunt me. All right, so it's on the green sheet, or protozoa. Giardia lamblia is a protozoa that's everywhere around here. And if any of you all ever got to spend time in a creek or a pond or anything, you probably had it. It can be self-limiting, and in a healthy person, it's probably not going to cause too much upset. But it's in all freshwater sources in this area. And it can actually be in wells as well. So it can be in people who have, how many of you have well water? No, what? Wow, that's more than I thought it was going to be. Okay, so I grew up with spring water, so I know we all had it. This little protozoa lives in the upper part of the small intestines near the duodenal region, the duodenum, and it can cause alternating diarrhea and or constipation, and it can cause like gassy. Yeah.
Is it only if you get it in your mouth or just like?
No, yeah. So you would like because you know you're swimming. Did y'all play creeks?
Yes.
Okay, so you know creeks and pond water and any lakes, any freshwater source here. So Giardia can live and it can cause upper stomach pain, like at this area, stomach pain. And then symptoms can kind of go away, and then they can come back and they can go away. But here's the thing: what they can do is they can do a stool culture, and they'll look for that, or they'll look for the cyst of that. But what they can also do is a string test, and I want y'all to have heard of this string test. And if you look at this, it doesn't take too many to get sick, does it? This protozoa lives in all wildlife's intestines, so raccoons and beaver and all this. It's part of their normal flora. We have protozoa as part of ours too, but this is a human pathogen. So if we get it and it doesn't take much, we can actually get sick from this, and it can last a while. So what people describe, and don't laugh when I say this, but this is the kind of thing you kind of want to remember. This is the test question they ask you on ridiculous test, but it can cause a foul-smelling stool. What? Okay, so you're like the only one that laughed at that, because which stools don't smell bad, right? But this causes a particularly foul-smelling stool. So it also can cause stools sometimes to take on a greenish appearance. So this is the protozoa that can do that. So what they can do is they can do a stool culture, but sometimes stool cultures can be falsely negative. So that it can be kind of cumbersome and expensive, and it can take, and nobody likes giving a stool sample, right? Because it's just like, okay, what am I supposed to do here? So anyway, but what they can also do is a string test, and I'm not sure this is any better. But the string test, and I have done many of these in my career, but what they will do with the string, they have this huge pill that's got a string wound up in it. So some of the part of the string is out, and they hold that out while the person swallows the pill, and the string is unwinding as they swallow it. And it will unwind, go through the stomach and to the first part of the small intestines, the duodenum. And they tape the string on so you don't swallow the whole thing, because then you can't test it until it comes out the other end, but you don't even want to then. But anyway, and they leave it in for 24 hours. And then guess what they do? Pull it out. So then they pull it out and send it to the lab, the micro lab. And this is really a better way to do Giardia, because what you do with the string then is you get all the good stuff off the string, and now you've got what's left, which looks a little bit mucousy. But you've got this in bio, and you look, and then the Giardia, you're going to really be able to see them, because that's where they live. They live there in that first part.
Wouldn't it track it up?
You pull the string. Yeah, you just pull it. It'll.
I would
Why would it dissolve like a regular pill?
Well, the pill just has this. The only reason you're swallowing the pill, which is just a little encased thing, that just ends up going on through you. But it's the string that you're delivering. So it's called a string test. This is a test that can be done for Giardia. Yeah. The Giardia?
No, the string.
No.
It's just some yarn.
It's just a string. You could actually swallow it. I mean, I swallow such worse, like marbles and stuff. I mean, and then I waited to die, because my mom told me, she said, take the marble out of your mouth. If you swallow it, you're going to die. So then, of course, I'm outside playing, and I swallow one. I obviously had a problem. But anyway, I swallowed one, and I didn't tell her, and I waited like 2 or 3 days, and finally I started to cry because I was sure I should have been dead by then. And so I was like, started to cry. She said, "What is wrong with you?" I said, "I swallowed a marble and waited to die." And she started laughing. So, and I'm thinking, I remember that very well. I remember exactly where it was when I swallowed it and realized I was going to die. Strings wouldn't hurt you at all to swallow. So
You can swallow the marble, you can swallow the string.
no, seriously, that reminds me of a story that I'm going to make y'all sit through. I don't think I've told you this. And it kind of goes along with immunology, which we're about to talk about too. You all, some of you know because you just had an too, and you understand that when you have an infection, your body's immune system mounts a response. And part of that response is to build what kind of a protein to protect you? Antibodies that will protect you by scavenging up and getting those foreign things and getting rid of them. And while you're mounting your antibody response, you have signs and symptoms of the disease. But as soon as you have enough antibodies, you start to feel better, don't you? So we call that a natural progression of disease, where we have the incubation for the disease, we have this prodromal phase, we have the infection, and then we convalesce, right? And in the meantime, that's happening because we get these antibody responses. Is everybody clear about that? So I'm going to tell you, I don't think I told y'all this, a true story of a physician that had a case, and in fact, he was an ID doc, so had a case of infection. They never knew what it was. They never got the ID, never did. Do you think that we always figure it out? No. We don't. But this little boy had come in and he was deathly ill. He was in ICU and he was deathly ill, and it was obviously an infectious process. And pretty much the parents had been told it was not likely he was going to make it. But they had no idea. They never got anything cultured, never got anything. And so, but the little boy turned a corner and started to get better against everything that they thought was going to happen. And got better. And it was really, I'm sure parents thinking this is a miracle, right? And so, whatever, the little boy got better, and they really didn't think he was going to. And then his little sister started to get sick. His baby sister started to get sick, would look like the same signs and symptoms, crashed just as quickly as he had. She's in ICU now, but he's recovering. So this ID doc and his team got together and said, he made it against every odd because his system was shutting down. Hers is at the same thing. And to think she's going to make it is a far stretch. But what we could maybe do, because they have the same blood type, is to take his plasma that has antibodies in it and give her plasmapheresis. Because he's made the antibodies. And it might treat her, this baby sister, who's dying. So the docs go to the parent. I didn't tell you all this, did I? The docs go to the parents and say, we don't know if this is going to work, but this is something that might work. Do we have your permission to do it? Because it means we need your son. We're going to get some plasma from your son, and we're going to do it. So the parents were like, of course. Let's try this, So they tell the little boy, and if I remember correctly, he was like 6 years old. And they tell the little boy, they said, because he's better now, he's recovering, he's in convalescence, period. And they told him, they said, your little sister is sick and she's not doing well, but to do better, we need your blood. And it might save your little sister's life. And the doc said that the little boy was like hesitated for just a few minutes and was looking at him and said, okay. And then, it took a few minutes, a few seconds really, but it seemed like a long time. And then the little boy said, how long will it take me to die? Because in his mind, he heard, we're going to take your blood and give it to your sister So she can live. Is that like one of the saddest things you've ever heard? Okay, so here's what I need y'all to know. When you talk to little kids and tell them they're going to die if they swallow a marble, or if you're going to take their blood and save somebody else, you might want to try to put yourself in their minds and let them know exactly. Yeah, just be a little bit more specific. But anyway, the little girl survived. It worked. It worked. It was really essentially the same thing as giving gamma globulin that we do. Never figured it out. Never figured it out. Yeah. So, and by the way, guys, you know, it's the only field of study or of a profession where you're expected to know it all, and it's impossible. Nobody does. And you won't always find out. You won't always know. But there's lots of opportunities to find out a lot. But look, like, so gonorrhea, woohoo, takes a little bit more for that. But hey, guys, thousands. Hey, listen, y'all know. No, okay, that's not even something different. It's bacterial too, Neisseria gonorrhea. But actually, it's 1,000 cells of a bacteria. A lot. Remember y'all's little touching a colony and doing it. Y'all were seeing thousands just on that. It doesn't take very much for that. But anyway, so this is just letting you know, you don't have to memorize this. I would never want you to. I just want you to understand for each infectious disease we do know about. We do know about their portals of entry. We know about their infectious dose, what it takes. And so we can use that in trying to help to prevent the spread of disease. Obviously, the ones that have very low ID numbers, then it's harder because it takes so few. We've already talked about some of the virulence factors and all of that. As far as how they cause disease, again, this is probably intuitive, but make sure you could kind of discuss it. There are 3 ways that microorganisms damage the host. They can do it directly through their own enzymes that they make. They make enzymes that end up tearing our tissues down, which is wicked, but you know, they can do that. They can make, some of them make toxins, and it's the toxins that are doing the damage. So that's, let me give you an example of ones that do that. Corynebacterium diphtheriae, y'all know what that causes? Diphtheria. Corynebacterium diphtheriae, it's a bacterium, it's in your little bacteria things. It causes diphtheria. The damage is being done by the toxins. Tetanus. Tetanus is another one. The damage is being done by the toxins Clostridium tetani produces. So why am I stressing that to you? Because it means that even though you know they have that, and you could treat because they're bacterial with the what? Is the antibiotic going to be the have all end all? No, because it's not even them. It's their toxins you have to get rid of. You have to give an antitoxin for the ones that you know that secrete toxins, if you've got one, and you might not always have one, but you've got to try to get an antitoxin for those. So endo and exotoxins. I don't know if I've told you yet, but let's go ahead and kind of know. The gram-positive organisms usually are the ones that give us exotoxins. So let me give you a couple of examples, like Clostridium tetani, that causes tetanus. Clostridium botulinum, Corynebacterium diphtheria, they're gram-positive. Endotoxins are usually going to be produced by gram-negative bacteria. Again, is this just mostly telling you that you need to know what you're dealing with? Yeah, so this is just to emphasize the importance of getting that correct diagnosis. So these are the first 2 you see, that's how they can cause. But sometimes they cause damage because they elicit such a response in your immune system that your immune system response is so over the top that it's your own immune system response that's causing the damage. Again, an incredibly important thing to know, because if you're dealing with a microbe that you know can do this, you've got to suppress that person's immune system. And that might be the treatment. And that sounds really counterintuitive because, Sarah, you would never want to suppress somebody's immune system intentionally. Yes, you would. If you know they've got an infection, that it's going to be the immune system response causing most of the damage. Yeah.
Can you say the gram-positive?
Gram-positive organisms mostly produce exotoxins, and gram-negative produce the endotoxins. And yeah, thank you for actually asking that, because maybe I'll also tell you something else. Let's see if this makes sense to you. If it's an exotoxin, how does that suggest to you these bacteria produce the toxin? And they make it and then they excrete it. So killing them after they've made it, you do want to treat them, but still the toxin's out there doing the damage. Okay, wait a minute though. An endotoxin, gram-negatives produce endotoxins. Where are those toxins located? Inside of those gram-negative bacteria. If that toxin's inside, Casey, inside that gram-negative bacteria, and you give them an antibiotic, what do you know you're going to be dealing with next? You don't know?
I'm so lost.
Okay. That's okay. I've been lost. I've been lost for a while. So it's okay. But the case endotoxin means that the toxins inside those bacteria, those gram-negative, like E. coli, Klebsiella pneumoniae, Proteus mirabilis, these gram-negative organisms, some of them produce endotoxins. The toxins are inside the bacteria. And you see that they have these infections, and you give them an antibiotic. And usually I can do that. But you give them an antibiotic. But when you give them the antibiotic, what happens to the bacteria? And when it splits open, what are you now dealing with? The toxins are now released into the system. So this is sometimes why when somebody gets a treatment for certain things, they get worse. They get worse after. Why? Because of the what? That you just released into the system. The endotoxins.
Gram-negative is endotoxin.
Yes. So gram-negatives bacteria are the ones we tend to think of that make endotoxins, and gram-positive bacteria are the ones that we think of that produce exotoxins, which are incredibly powerful. But we do have the ability to also treat with antitoxins. So we don't just have antibiotics as chemotherapeutics, we also have antitoxins. I'll give you a real case scenario that my son was just got to see. It wasn't very good for the family or the baby, but a baby that had botulism in Roanoke, which we rarely see Clostridium botulinum causing botulism. Now, we rarely see it because of the FDA and because of food, directives and whatever. We don't see it that much anymore. It causes a flaccid paralysis. So if babies become, they call it floppy baby syndrome, the babies become like flaccid, they can't contract. What's it going to eventually affect? Which muscles? The muscles for what? Yeah, the muscles for breathing. So this is obviously a serious situation, and without treatment, death, period. But this is caused from Clostridium botulinum, a bacterium that produces a what? A powerful exotoxin, botulinum toxin. We call it Botox. We actually can use it for treatment of some things, right? It's so powerful. But for this baby, there has been such few cases of it that the antitoxin that they had to get was from California. And to get it from California to Roanoke took a few hours. And were the hours making a difference? Yeah, but that was the closest that they could get called, which I thought was kind of absurd. I couldn't even believe that it was taken, but the baby turned out okay.
So is it quick onset then?
What?
Is it quick onset or slow?
Plus the botulinum
Yeah.
disease is pretty quick onset. Yeah, and so, but this is why they tell people, they tell you not to use organic honey. Maybe, I don't know, y'all probably were given pacifiers dipped in honey, weren't you? I mean, maybe, I don't know. Whiskey? Okay.
There you go. There you go.
So, that's an old thing that people did. But anyway, they even 20 years ago said pediatricians tell you, do not put honey in oatmeal. When you start doing oatmeal, don't put natural honeys in things. It's okay when they get a little bit older. It really can be okay sometimes. because natural foods in your ecosystem can also help to colonize you and to protect you. But little babies should not be given those types of things because it can really overwhelm.
Organic is not good for babies.
Well, don't quote me saying organic's not good. I'm talking about honey, because organic honey is out there in the environment, and Clostridium botulinum is out there in the environment. So specifically, no, it's not because of botulism. But since we stopped those practices, we rarely see it anymore. It used to be pretty common that babies would die from, it's called infant botulism. Y'all can look that up.
Yeah, like we bought honey from, there was like a thing, I lived across the fire department, so they like have a marathon. And this dude was selling honey, and he was printing labels on the honey not to give to babies or children until like they're over 2 or 3.
Yeah, exactly. And it's for that reason. Because there can be spores. It only takes spores to be present in the honey. It doesn't actually have to take living organisms. The spores can live through pretty harsh things. But anyway, so we have antibiotics to treat bacteria, but we have antitoxins to give for the ones that produce toxins. And then sometimes we suppress a person's immune system, which sounds so counterintuitive, but you have to, because in some diseases, it's the immune system that's causing problems. Okay, so some of the enzymes we said that some bacteria produce that can actually cause damage. You can see, like, and I'm not going to ask you any of these. Y'all know they're enzymes because in ASE, and I won't ask these. This is kind of FYI, but you can see what they do. So mucinase, ACE, affecting the mucus things. Did we say mucus is good or bad in our systems? It's good, isn't it? We really don't want somebody interfering with our mucus coating, because right underneath that mucus coating is the bloodstream. Right? So we don't want anything getting into that. Same thing with this enzyme that breaks down hyaluronic acid. And that's holding our cells together, keeping the integrity of our joints at what they need to be, that kind of thing. So anyway, you can just see some of them produce enzymes, and the ones that produce those, hemolytic enzymes, coagulase, those types of enzymes, then they can become, they're the ones that are the most virulent. When we think about toxins, would you all recognize from prefixes what they act on? Neurotoxins are going to be toxic to the what? Enterotoxins to the intestines. Enteric means intestines, right? So enterotoxins. Nephrotoxins to the, and you would want to know this. Hemolysins are going to interrupt our blood cells. So these are just basic definitions. I've shown you all, and you are going to see on Monday from your own plates, you're going to see that on a sheep blood agar plate, some microbes, like the microbes are right here, but are they sending out hemolytic enzymes here and lysing all of this? This is called complete or beta hemolysis. You don't need to know that, but that's a complete beta hemolysis. This is an alpha partial hemolysis. And some aren't hemolytic, but we know that some are, and so that can contribute to that. Anyway, so you see some of these gram negatives that are actually, and I just don't, that's fine. You don't have to know examples, just the gram negative endotoxin, typically what we think of, and gram positives or exotoxins. Okay, so this just is a little characteristics of some of them for more information if you want to look at that. And then let's see. And then we said we get this idea that tissues can die prematurely. Pathological death to a tissue is called necrosis. And if there's been pathological death to a tissue, this is obviously a serious kind of thing. And sometimes it's going to mean that the tissues you need to help them and get them removed. Yes, Jared.
I'm sorry, could you go back to your types of toxins? I didn't quite get them all that.
So it's on slide 22 on PowerPoints, but it's on slide 22. So neuro, entero, nephro, and hemo. Really, hemolysins, which are just going to be enzymes that end up doing that. But these are the things they can do. Those prefixes tell you what they're targeting.
Okay, I just didn't get the spelling of the last one. So
Okay.
Thank you very much.
You're welcome. All right, so so we said that you could look at that just for more information if you wanted it. And This is something, again, these are kind of just terminology, but I think it's important that you've heard of them and that you know. So definitions for infection types. If something's localized, what does that describe to you? It's one area. If something is systemic, so which would you choose if you got to have one? Yeah. So can localized become systemic? Yes. So we do know that. So infections that spread from a local site that have become systemic, these are called focal infections. Mixed means what? Polymicrobial would simply, poly means what? Many microbes. Now, luckily, there aren't that many things that are caused from polymicrobial infections. But you all will see in your study of chapter 10, when you're looking at treatment, that when there might be a polymicrobial infection, you can imagine that the treatment becomes a little bit more complex. And you might have to do like extended time on the treatment, so a prolonged dose, or you may have to do combination therapy. So a combination therapy to make sure you're hitting any of the microbes that could be causing infection. Can you have secondary infections? You know, I think I've already mentioned to you all that most pneumonias are caused from what?
You're already sick with something.
What'd you say?
You're already sick with something else, and then that's.
Oh, no. So most pneumonias are caused from viruses. The most pneumonias are caused, if I hadn't told you all that, let me tell you that now. Most pneumonias are caused from viruses. Almost, yeah, just by far and away. I don't know the actual percentage, but by far and away. But why for every pneumonia that we know, if somebody tells you you had pneumonia and they weren't in the hospital on IV antibiotics, it was viral. Why do you hear them say they would put on an antibiotic? You do it prophylactically, meaning you do it to prevent a what? A secondary bacterial infection to cause the pneumonia. When somebody has viral pneumonia, most of those people would actually get better on their own. They might feel like hell while they're getting better on their own, but most of them would. But some of them, because of their suppressed state, will get a secondary bacterial infection on top of it, and that is deadly. A bacterial pneumonia is deadly even with medical treatment. So the reason that everybody gets an antibiotic that has pneumonia, when we know that they're viral, is to prevent the secondary. Are you with me? That's just an example. I don't even know what example they give you. What example did they give you? Oh, okay. So like even people who are compromised that have influenza, y'all know influenza is caused from what? A virus. You don't treat an influenza with antibiotic, do you? No, you do not. Not usually. Unless you got a person who is going to be in a susceptible group that the influenza is going to make them sick enough that you're worried about a secondary infection, and then you are going to give them an antibiotic. And y'all know me. I tell people don't treat anybody for anything. No, I don't say that. I don't. I really know that all these things save lives. But I do think that that's absolutely necessary. If you've got somebody that's in that group, very young, very old, they have a viral influenza. Yeah, absolutely give them an antibiotic too to prevent that secondary thing.
For like the human bite infections, is that like most common if like a baby accidentally bites you or something? For the human bite infection.
Is that up here?
Mm-hmm.
Where is it?
Under
Oh, I see. Okay, so mixed. Oh, yeah, so human bite. Yeah, because what do we know about your mouth?
It's gross.
It's not. It's not gross. Our microbes keep us healthy. Our microbes keep us safe, right? Our microbes keep us healthy and safe, but they're really not supposed to be embedded into your soft tissue, of your skin. So when somebody bites you, and it's human or animal, when somebody bites you, they're introducing these microbes that are now in a foreign place, and these microbes can take hold. And it's more than one. It's several. So this can be one of those times that is perfectly acceptable to treat with a combination therapy for maybe a prolonged period of time, just to make sure that somebody doesn't end up getting, right?
Is that skin is broken?
Yeah, I mean, I would say But you might see somebody who thinks, you know, I can't see if the skin's broken, but it might be broken, so let's just be safer than sorry. But I would say it would need that. Yeah, definitely. So, thank you. Good observation. Acute infection, y'all know, comes on quickly, and the symptoms are usually pretty serious, aren't they? You know, it's like that 1 minute you're well, and the next minute you're thinking, am I nauseous? And the next minute you're vomiting, right? Or diarrhea, or both at the same time. So it can come on really quickly. And then some are chronic, that you know, the symptoms kind of are vague, and maybe you kind of come and go, but they're going to last typically a long time. So anyway, this is just some of these. I've already given you definitions for signs and symptoms, haven't I? Y'all know this. A syndrome. If you don't have this, make sure you do. A syndrome, a disease identified by certain signs and symptoms. And so if you have enough of them, like, you know, for some syndromes, like Kawasaki syndrome, this is one that if you have, like, I think they say 3 out of the 5, they're going to say that you probably have this syndrome. But anyway, so every syndrome is different in how they want to see it. But they've seen them enough that if they have enough signs and symptoms, they say, yeah, it's really likely that you're in this syndrome. All right. And so again, the symptoms are things that, signs are things that you can measure. Now, I really hate this, because can't you measure fever? So when you see something that makes sense to you, question it, because that's not really. Yeah, so I don't know. Ignore this thing from your book. Ignore it, because that's not true. If you can see something and measure it, it's a sign. If it's something that you can't measure, somebody's just telling you, like about pain. I was just reading something that somebody said about pain, and they had a patient that said their pain level was at 10 when they got their blood pressure cuff done. Like a blood pressure cuff, the pain was at 10. Where this person was saying they'd seen people in their ER that would have like almost amputations, and they would say their pain on a scale of 1 to 10 was like a 3. And they had literally like appendages hanging, right? So, no, I'm telling you what I read from this clinician saying this. So pain is a hard thing, isn't it? It's a really hard thing. We don't know. And everybody, and it doesn't mean that person who was getting their blood pressure, the blood pressure cuff wasn't, it might have really been higher. We're not going to judge. No judgment. If you ask somebody to rate something and they rate it, that's what they rated it. Okay. So I mean, you write it down. That's what you write down. They said. So anyway, so we know some. We've talked about subclinical, asymptomatic. We talked about those terms, and so y'all know that some things we know are that way. What do we know about many sexually transmitted diseases? They're what? Sexually transmitted. Well, we know that because that's what. Would you say? They're asymptomatic. Most are asymptomatic. That's why responsible people, once they become sexually active, get routinely tested on a routine basis, because we know that most are asymptomatic. So, all right, and then we know certain ways that certain microbes are spread. So some things, some fluids are not going to be dangerous, but like we said, saliva with HIV, but other fluids are going to be really have a lot of the organisms, so enough that it can lead to an infectious dose. Do we know this term sequelae? We do, don't we? And latency. So if you didn't know it, make sure you do know it. Latency means that these infections will stay in your body dormantly. And I think we talked about it when we talked about varicella zoster and herpes simplex. What did we say? Do you ever get rid of those? No, they stay in your system. And most of the time they'll be latent, and will they ever come out and cause you any trouble again? Most of the time not, but they can, right? So you can get shingles with varicella zoster. That's the sequelae, if you will. So definitely. And you guys, Lyme disease. Y'all know what causes Lyme disease? Well, a tick bite doesn't cause Lyme disease. And so thank you so much for saying that. Thank you so much for saying that, because that's what everybody would say. But ticks can bite you from now until kingdom come, whatever that means. I used to worry about what that meant. Back in the day, I worried about it. Look, but ticks can bite you all day long and you not get Lyme disease. Tick bites don't cause Lyme disease. Ticks are vectors of what can cause Lyme disease. What causes Lyme disease? Borrelia burgdorferi. Borrelia burgdorferi. Thank you, Jared. And it's a bacterium, isn't it? It's a spirochete. It's a bacterium. Borrelia burgdorferi is what causes Lyme disease. And it is a bacterium. So the good news, what is it? Good news, Brittany? Antibiotics, if it's treated early. And sometimes you miss that early inoculation, that early period of signs and symptoms. So when you had missed those, what can it lead to? Y'all all know this about Lyme disease, right? Y'all have heard this, that Lyme disease can lead later to like arthritis. And what does arthritis mean?
Inflammation of the joints.
Inflammation of the joints. That's what it means. Inflammation of the joints is not a diagnosis. So somebody has arthritis, and they're chronic, and they in that category of thinking, you know, you look like you hike, or you look like you hunt, or you look like you spend time outside. Good for you, because people are supposed to be outside. You might want to think about ruling that out. You might want to do blood tests that could rule that out. Lyme disease.
So when you say vector, they're just like a carrier?
Yes. So they're a carrier. So when y'all say like Rocky Mountain spotted fever, tick fever, it's really not the ticks that are causing it. They're the vectors that are bringing it to you, right? And that's a different. I know that sounds like a splitting of hairs. It's not though. It's these microbes that causes polio. So polio, this virus that causes polio, it kills all itself in that initial disease. It has a significant mortality rate. But even people who survive that, can they have long-lasting paralysis forevermore? Yeah. So these are some of those things. Meningitis. We have seen the numbers of meningitis go down so wonderfully in this country. Why? Because of immunizations. We have the pneumovax that protects against Streptococcus pneumoniae. We have the meningovax that protects against Neisseria meningitidis. We have the Hib vaccine, HIV, that protects against Haemophilus influenza serotype B. We've seen the numbers go down tremendously. But before these immunizations, and I was part of this before all of those, I was in medicine already before those, it was common to have our ICUs filled with at least some at any given time that were trying to fight for their life. And then the ones that actually did survive having permanent neurological consequences like deafness that could happen. That was going to be fun.
My grandma's brother was like that.
Really? Your grandma's brother survived it, but he was deaf?
Yeah, it didn't happen till later, but when they were kids, he had polio.
Oh, he had polio.
Mm-hmm.
Oh.
And then now, like, it's been a couple of years, but when they talk on the phone, it's like it's not really talking. Like, he knows who he called, but my grandma can't barely understand him because he can't hear himself. That makes sense.
Yeah. Yeah, no, it does. It does.
Yeah.
And actually, there's something called post-polio syndrome that we're really seeing in really high numbers right now. And it would have been people most of the time because we had good coverage for like y'all's age group. People were getting because they remember the parents that remembered people like did you say it's your great uncle or uncle? Would you say who you say it was?
It's my grandma's
It's your
brother.
your grandma's brother, so your great uncle. So they remembered, they knew polio, they were scared to death, so they got their kids, they got all their kids immunized, right? But the ones that had survived it during those outbreaks that had been in our country then are now experiencing as older people what's called post-polio syndrome. And I think, we may mention it again, but just so y'all are aware of that. So having a good history on a patient to know if they've had something like that would be really important because sometimes the symptoms can seem kind of vague, neurological symptoms that seem vague, and they can be discounted as just somebody getting older, Don't discount old people. That's all I'm saying. Get a good history. Make sure you know what you might be dealing with, and because some of the times it can be helped. We gotta go, don't we? So look, if somebody will get out the back to incinerators and get those turned on, that would be really great. Will she let you come and do your cultures? Because I want you to do your cultures. Can I ask her if you can do 10 minutes? Will you be upset?